Good morning. Today's class is on Introduction to Parasitology. How do you define parasitology? It is study of animal parasites which infect and produce disease in humans. These parasites can be classified into two types. One is an ectoparasite, the other one is an endoparasite. The ectoparasites are those which inhabit the surface of the body of the host without penetrating into the tissue. Example, and usually these act as vectors for transmission of microbes. Example is Sarcoptes scabi, which causes scabies. The endoparasite, these are the ones which live within the body of the host, example leishmania, and any invasion by these endoparasites is called as infection. What are the different types of endoparasites which are there? These are obligate, facultative, accidental, aberrant or wandering endoparasites. The obligate parasites are those that cannot exist without a parasitic life in the host. Example, Plasmodium. Facultative, these can live a parasitic life or a free living life when the opportunity arises. Example is Naegleria and Acanthamoeba. The accidental hosts are those where they infect an unusual host. Example, the dog tapeworm infection, or in other words, Echinococcus granulosus, which infects the humans accidentally. Aberrant or wandering hosts, these infect a host where they cannot live or develop further. Example, Toxoplasma in humans. The host. The host is defined as an organism which harbors the parasite and provides nourishment and shelter. What are the different types of host? Definitive host. Definitive host is a host in which the adult parasites replicate sexually. Example, Anopheles mosquitoes for malaria. Intermediate hosts are those hosts in which the parasites undergo asexual multiplication. For example, in malarial parasite cycle, if you see, the humans are the intermediate hosts. Sometimes some of these parasites may require two intermediate hosts. For example, Facialopsis, wherein you have the first intermediate host is an snail and the second one is an aquatic plant. Reservoir hosts are those which harbor the parasites and they serve as an important source of infection to other susceptible hosts. For example, dog is a reservoir host of dog tapeworm or Echinococcus granulosus. Excuse me. Paratenic host is a host in which the parasite lives but it cannot develop any further and it may not be essential for its life cycle also. For example, freshwater prawn or crab for Angiostrongyloides catanensis. To give another example, if you see the freshwater fish for the Diphyllobothrum latum, wherein it develops into a pleurocercoid and only acts as a source, which are taken up by the cyclops. Now, what is this host-parasite relationship? If you see, there are three different types of relationships. One is the symbiosis, commensalism, and parasitism. So, what is symbiosis? This is a close association between the host and the parasite. Both are interdependent upon each other and one cannot live without the help of the other. None of them suffer any harm from each other. Commensalism is an association in which the parasite only derives the benefit without causing any injury to the host. Parasitism This is an association in which the parasite derives benefit from the host and also causes some injury to the host. The host gets no benefit in return. So, when it comes to the transmission of the parasites, it depends on what is the source of infection and what is the mode of infection. So, what are all the sources of infection for parasitic infections? Man. Man is a source or a reservoir for majority of these parasitic infections. If you see amoebiasis and if you see the helminthic infections, majority of the helminthic infections are through man, and man acts as a main source of infection. For example, a roundworm, hookworm, Enteropius vermicularis, all these man acts as a source of infection. Animal. The infection which is transmitted from infected animals to humans is called as zoonosis. Right? So, the examples of zoonotic diseases is echinococcus from dogs and toxoplasmosis from cats. Vectors. These vectors are, vector is an agent usually which is an arthropod that transmits the infection from one infected human being to another. You have two types of vectors, one is a biological vector, another one is a mechanical vector. A biological vector is one wherein it is required for the transmission of the disease and the cycle takes place within it. So, part of the life cycle of the parasite will take place within the arthropod. Such a arthropod or a vector is called as biological vector. What is a mechanical vector? Mechanical vector is the one which just carries the infective material from one place to another place. So, this is called as a mechanical vector. Contaminated water and soil. Soil polluted with human excreta, these will contain a large number of eggs or the larval forms of the parasite which can infect the man. Examples are hookworm, ascaris, strongyloides, trichuris. All these are through contaminated soil and water. Water contamination examples are cysts of Entamoeba histolytica and Giardia. The other source could be raw meat or undercooked meat. Examples of these are your tinea infections, wherein you have Cysticercus cellulosae from the pork and Cysticercus bovis from the cattle. What are the modes of transmission? Most frequent mode of transmission is a fecal-oral route. Infection is transmitted orally by ingestion of food, water or vegetables which may be contaminated with the feces which contain the eggs or the larval forms of the or the infective forms or infective stages of the parasites. Example, ascaris, cysts of Entamoeba histolytica, etc. The second mode of infection could be penetration through skin. Thus, the infection is transmitted by penetration of the larval forms of the parasites through unbroken skin. For example, the filariform larvae of Strongyloides stercoralis and the hookworm. These penetrates through the skin of the individual when they walk barefooted over the fecal contaminated soil. Or they can also be introduced into the human body through blood-sucking insect vectors, for example, Plasmodium species, Leishmania species, and your Filariasis or Ochraria bancrofti. Some of the parasites can also be transmitted through sexual contact. The most common example is your Trichomonas vaginalis. There are few cases recorded where Entamoeba giardia are also known to be transmitted through sexual contact. Usually this occurs among homosexuals. Bite of the vector. Many of the parasitic infections are transmitted by the insect bite. Example, malaria, filariasis, leishmania through sandfly, etc. Vertical transmission, that is from mother to fetus transmission can occur, and most important example is your Toxoplasma gondii, and sometimes even Plasmodium is known to occur. Any blood-borne parasitic infections can be carried from the mother to the fetus. The other mode is blood transfusion. Certain parasites like the Plasmodium, Babesia and Toxoplasma species, Leishmania, Trypanosoma, all these can be transmitted through transfusion of blood or blood products. Lastly, you have autoinfection. Few of the intestinal parasites may be transmitted by the same person, which may be through the contaminated hand or by reverse peristalsis. It is usually commonly seen in Taenia solium, Enterobius vermicularis. Now, coming to the life cycles of parasites, this may be a direct, which is a simple one, or it could be an indirect, complex one. The direct life cycle or the simple life cycle requires only one host for its whole life cycle. Example, you have among the protozoa you have Entamoeba histolytica, Giardia, Trichomonas, etc. Whereas among the helminths you have the cystodes like Hammerlipsis nana, nematodes. Most of your nematodes, for example, Ascaris, hookworm, Enterobius, Trichuris, Strongyloides, all these have man as a single source of infection, and the whole of the life cycle takes place only in man. Indirect, this requires 2 or more hosts. Man can act as a definitive host in case of Leishmania, Taenia, Filaria, Dracunculus, etc. He can act as an intermediate host in Plasmodium, Toxoplasma, Echinococcus. In case of indirect cycles, this will require 2 or more. Examples are Diphyllobothrum, Fasciola, Paragonimus and Clonorchus. So these are the examples where you have more than 2 hosts are required for the life cycle to be complete. Now coming to the pathogenesis, usually there are various methods by which the parasite can cause the disease or the changes within the body. For example, you have the mechanical trauma. The eggs, which are large in size, can be deposited within the intestinal mucosa. For example, Schistosoma mansonae, or it can be deposited within the urinary bladder. For example, Schistosoma hematobium, or in the lungs, Paragonimus. Okay, so all these can cause mechanical trauma. The second pathogenesis could be a space-occupying lesion. For example, the hydatid cyst, or otherwise the Echinococcus, wherein you have the cyst which is large and may compress the surrounding tissues or organs. Inflammatory reactions. Most parasites induce a cellular proliferation and infiltration at the site of their multiplication. Some may produce enzymes and lytic necrosis and bring about the pathogenesis. For example, intracellular parasites like Plasmodium, Leishmania, Trypanosoma, etc. produce several enzymes which cause digestion and necrosis of the host cells. Some may produce toxins, for example, Entamoeba histolytica. Majority of the time it is an allergic manifestation because many metabolic and excretory products of the parasites, they get absorbed into the circulation and produce a variety of allergic manifestations in the sensitized hosts. Example, if you see Schistosoma causing cercarial dermatitis. Lastly, there can be neoplasia. Some of these infections parasitic infections can contribute to development of neoplasia. Examples are Schistosoma hematobium, which causes bladder carcinoma, Clonorchis, which causes cholangiocarcinoma. Host immunity. The host immunity depends on it can either be innate immunity or acquired immunity. Innate immunity depends on the age, sex, genetic constitution, skin barrier or gastric barriers. If you see the age, both extremes of age are more vulnerable for parasitic infections. To give examples of infections which are common in males, for example, you have amoebiasis, which is much more common in males than compared to females. Females develop are more vulnerable to the hookworm infections because they develop anemia. To develop anemia due to hookworm infections. Then skin barrier acts as a wonderful innate immunity, and the gastric barrier, because most of these parasitic eggs are susceptible to acidic atmosphere. Acquired immunity, you may have humoral or cell-mediated immunity. Most often it is a cell-mediated immunity. Coming to the laboratory diagnosis of the parasitic infections, majority of the time it is a microscopic or macroscopic examination of the stool sample which can reveal the presence of the parasitic infections. If you see worms, for example, you have the worms which can be passed out in the stool, for example, your roundworm, pinworm. Then from the intestine, you can also get the worms like Physiolopsis. All these can be macroscopically visualized. The presence of the ova or the larval forms in the stool sample can be observed by doing a microscopic examination of the stool sample. Sometimes culture can be done. For example, RPMI 1640 is a medium which is used for cultivation of malarial parasites. You can use NNN medium for cultivation of leishmania. Then you can have immunodiagnostic methods where you can detect the antigen and antibody of the parasites can be detected. Molecular methods can be used for, say for example, if you are having entamoeba histolytica, to differentiate between entamoeba histolytica cyst and the entamoeba dispar cyst, which is just a commensal, you may have to do a molecular method. Xenodiagnostic techniques can also be used, animal inoculation and imaging modalities. To say example for imaging modalities, hydatid cyst is one of the best example you could give, or for filariasis, for filarial dance, when you do an imaging method, you can pick it up and can diagnose the condition. Which we will tell you all in the laboratory diagnosis. Individually, when we take the parasites, we shall give you more in detail. Treatment, these are some of the antiparasitic drugs which are used: metronidazole, chloroquine, paramomycin, cotrimoxazole, praziquantel, niclosamide, albendazole. All these, praziquantel, albendazole, mebendazole, all these are most often used for your helminthic infections. Ivermectin, diethylcarbazine, all these are the some of the common antiparasitic drugs which are used for treating the parasitic infections.